I'm basically talking about the retina inside the eye and how it relates to vision. Now, I'm not going to talk about anything having to do with eye operations and surgery. That's not my domain.
Hold straight.
Yeah, so
Yeah, there we go.
you'll see that in a second. So, I guess to start with, I'll give you a little quick background. I was born in Chicago. We moved to Elkhart, Indiana. When I was a kid, I grew up there. To my great benefit, it was an excellent, excellent town. And then I went to Northwestern University on a scholarship as my first foray into higher education. And it was there that I met my wife, Lucy, who's over here with her head down. And we met in a comparative anatomy course, actually. Amongst other things, we worked on the dogfish shark, which is a strange place to start. It also fits in. So after Northwestern, then I went to optometry school at Indiana University in Bloomington, which was one of the leading optometry schools, and got a very good education. Actually, previously at Northwestern, I'd been active in fraternities, and all my fraternity brothers, had they survived, never would have believed that I ended up being a serious scientist for 50 years. But in any event, I went to Indiana Optometry School, which was a very, very strong program, and that's where I got my basic interest in the retina and the eye itself. And that interest got to be so strong that I did further graduate work. So I got a Doctor of Optometry degree at Indiana. Then I was accepted at Brown University in the Ivy League for graduate school, and I went there and got my PhD there with a lot of emphasis on the retina. And actually at the time, this is a long time ago, of course, Brown was a handful of places in the entire country or the world where you could do this sort of research on the electrical signals of the eye. And that's where we're basically going today to see how that worked out. So then after that, I went to the Johns Hopkins University Medical School for advanced training, where I got further work having to do with the retina. And also there was at Hopkins at the time, there was a group of 4 people who were very, very far advanced in the field. We were sort of a cadre, and we sort of become, this was back about 1960, maybe 66 probably. And this cadre of us sort of dominated much of the field thereafter, because as you'll see in just a few minutes, this is a highly specialized approach. So then left Hopkins, and I got a faculty appointment here at the University of Minnesota. That was in 1968, I believe. And I stayed there for my entire career, which went for 42 years, and it was an exceedingly fertile environment. Also at that time, Minnesota, again, was one of a handful of schools that had advanced programs of my sort. So it was a very unique setting, and it was very compatible for me by and large. I was in a psychology department, which at first glance sounds rather confusing, but I won't try to clear that up until we get further along. So my talk then has to do with the retina, which is often referred to as the brain in the eye, little brain in the eye. And you may also remember that Shakespeare said famously about the vision in the eye, the miracle of the eye. And for those of us who've worked in it for many, many years into the modern era, long after Shakespeare, it's turned out to be a much bigger miracle than even he thought. So I've been involved in for 42 years in terms of trying to uncover some of the basic things that are going on there. And I'm going to try to give you a simplified version in the next couple of minutes of what we did, why we did it. And this is where it sort of starts. This is a section of the eyeball. Whoops, what I don't want to do here. Funny. So we're talking, the retina then is a very deceptive sort of thing. This, of course, is a classic view of the eyeball. The inside of the eyeball has a very thin layer of nerve cells, and that's where most of the action occurs, to the surprise of anyone. This is a very thin membrane. It's about 1/4 of a millimeter, and it's also somewhat unstable, and you would think that the early anatomists who tried to look at it would have discarded this tissue at all. It's like a wet piece of tissue paper, and you would think this doesn't have anything to do with version. Let's go deeper into the eye and see if we can find what's happening. Well, it turns out this is where things really happen, as I'll try to show you as we go on. Let me back up for a second about the retina. And as I was touching on the anatomy, it was in the first thing that occurred understanding this was using an ordinary light microscope, or very special light microscopes. And this started in about the 1850s with the Germans quite active, but at 1900, an Italian by the name of Golgi and a Spaniard by the name of Cajal did fundamental research on the anatomy, and they were able to find things that nobody had seen before. And this then developed, so the first part of our story actually has to do with the anatomy and to find out what in fact is in this thing called the retina, this fine tissue paper. And as it shows up here also, you see that it's only about 1/4 of a millimeter thick, and it has about literally millions of nerve cells, if you can believe it, packed into this very, very thin structure. And our goal as we'll go along is to try to understand what's going on in those nerve cells. Now, over the years, the anatomists were the first to make big improvements, insight into the retina. This goes back to about 1960 now, and it's also a human retina, where it was possible to preserve the tissue very well in a very, very thin section, which allows you to see all these details. And the 2 points really to come on this, unfortunately, I lost my pointer, so forgive me if I'm looking for stuff. The structure is unbelievably clear and ordered, as you can now sort of see in this slide. The very top of this retina contains the receptors for light, and these are known as the rods and cones, which probably many of you have heard of before. And it was discovered indirectly much earlier that the rods were responsible for night vision, and the cones were responsible for color vision, as I'm sure many of you will remember. Now, the big point of this is that we're still talking about just the anatomy, seeing what the cells are. And we don't know in many ways exactly what they do, and that's the big goal of actually my research and many others, starting in about 1968 or so. Now, this is a schematic view of the retina, and I'm not going to go into details, but I want to make it just to make the point that at the very top there in the purple, these are the rods and cones, but then there are many other classes of cells connected to these rods and cones, and this represents then sort of a highly schematic view of what the retina would be viewed at by a modern anatomist. Again, it's very schematic. It's just a drawing. It's not the way they would look in the real world, but this will give you some perspective as to what we're doing. Now, once the retina was discovered, it was realized that there were sort of three main layers in this retina, starting with the receptor cells at the top, that's the rods and cones. Then they come down to cells known as the bipolars and ganglion cells. And this is a highly, highly simplified view of the retina, but it's sufficient for what I want to do today and not show you things that are impossibly complicated. Let me back up just for a second. So these are the main cells of the retina. And the object has always been to know what these cells are really doing. You can't understand vision until you understand what these cells are doing. In the early days, it was realized that the retina generated an electrical signal, which could be recorded if you put a contact with the external eye, and a small voltage would come up, which could be recorded. This was known as the ERG and was used for many, many years. It's very, very much like the EKG, where electrodes are put on the chest, but they're not on the brain itself. They're just getting this current that comes through the tissue. So this ERG was useful, but it could not answer this question of what are these cells doing? And that's where, very shortly now, I get more actively into the picture. The big breakthrough here was done by a Japanese by the name of Tamida, who in Japan at the time had developed something that became known as the glass microelectrode. And this was an amazing, amazing tool that he developed, which opened up this field almost completely, as I'll try to show you as we go along. Now, this glass microelectrode is a strange, strange piece. It's a hollow piece of glass, like many of you have seen in a high school chemistry course. And many of you have seen that if you heat glass, a tube like this, you can pull it apart. That's basically how this glass microelectrode was produced, but in a very, very special way. It's a microelectrode because it's very, very, very small at the tip. So if we concentrate here on the tip, this is what eventually, as I'll show in a couple minutes, we can actually use as a probe to get inside a neuron, a retinal cell, and see how it's really responding. But it's a remarkable, remarkable device in the miniature. This is the tip is shown there. The tip comes to a very small tip, but the point is that the glass's tube is still slightly open, and that means it can be used to record electrical activity. And so if we explode this just a little bit, this is the tip, and down there we're showing a magnified view, and the text down here, if you can read it, says the tip is about 1/100th of the size of a retinal neuron. So it's super, super small. It's really micro is the way to think about this. And many people thought that it would never work because it was much too small to use for electrical purposes. But it turned out that it did work fine well as it was perfected. Now you need a microelectrode, and you need somebody with a lot of understanding of the optics of light, and somebody who knows their electrical engineering, and somebody also knows how to harness these signals. And this is a photo now from my lab, and as some of you will notice, it's many years ago. But it's still, it worked. And I kept this lab going for 40 years at the U, and it has the parts of it over here on the left, as you're looking at it, this part contains all the devices used to actually control the light. We're going to take a light flash and put it down on the Retina and record the results. But in this slide, this part of the lab on the left side, there's a whole bench here consisting of many lenses and mirrors and optical elements just to produce a very small control of the light. That then goes into an electrical shielded cage, which is in the center of this. And it's an electrical shielded cage because the signals that we record, as I'll show you in a few minutes, are still very small, and we can't just have this thing sitting out in room temperature, in the air, although we'll see all sorts of electrical interference and never able to see the response of these neurons. So that's what the cage is for. Then there's a host of electronics over here at the right, which are used to process the signals that we record, and also displayed finally on an oscilloscope, where we may eventually see something like the display over here at the right. I'll quickly mention that this stuff, you cannot go to your Sears Roebuck and buy it. And even the scientific suppliers do not produce this sort of thing. It's put together by the investigators, and my background in optics was very helpful in doing this. And people even who came to my lab over the years, they always marveled at the complexity of my opticals on this bench. So anyway, that's the setting. This almost looks like a cockpit in an airplane. It's sort of relevant. We got stuff all around us. Normally we close this cage so that we can get rid of electrical interference. Okay, that's the view I want to see. So now we've got this microelectrode that is a micro, and we've got an operator who supposedly knows what he's doing. That's me. And without me, there doesn't matter if you've got a microelectrode, because nobody knows how to use them. We had a cadre. It was the golden era of this research. We probably had, originally we had 4 people that came from Hopkins, and I came to Minnesota, and one went to Berkeley, and one went to Columbia. And they were sort of the cadre of this group to start with. Then they trained students, but there were still only a handful of universities in the country, and none in Europe, who could do this sort of work. So it was highly, highly specialized. Now, so that's basically where we are, and now we're about to see what actually occurs in these cells by using these microelectrodes. Okay. Now the first thing shown here is this is a response of a cone. Let's see if we can back up very quickly. See if we sort of what we're doing here. Okay, that's the retinal structure. Now at the very top we have these rods and cones, and the big question to start with is, what are they doing? How do they work? And there were hints from visual scientists over the years, but we never knew directly what was happening. And even when we recorded up here on the exterior of the eye, it was very unclear how the retina really works or what was happening. So the good news is that that's where I got involved for many years, and we actually did discover a few things you'll be happy to know. Now this is the first thing to make this simple. This is the electrical response that you get from an individual cone cell. You put a flash of light on the retina, and then you record the voltage that is generated by the cone. That's what the upper trace shows you. I'm sorry I can't use mine. I don't know why our don't know how we can get back. We can't. So the top at the top, we're seeing the response of a cone, and at the bottom you see a little sort of square. That's when we put on the white flash. We put it on for a half a second, and then at the top, the cone response as you see it there, and it generates a very big response in this case because this is intense light. If we use a weak light, then the response gets smaller and smaller. And this was a big surprise in the understanding of cones. Now, this glass electrode that I mentioned, I think I did say, is hollow at the tip. And as a result, we can actually, to use it as a device to record electricity, we have to fill it with a strong salt solution. But the good news is it's glass, therefore these nerve cells will tolerate the glass and will even tolerate the electrode put inside the nerve cell, and that's the big deal. This is usually called intracellular recording because it's inside the cell. We have to get inside the cell to see these signals, and that's another interesting but long story as to how we do this, because it's not straightforward, and it doesn't work every day either. As my wife Lucy will tell you, many days I come home during my career all downcast because nothing happened. We never found these cells. But on the good days, and that kept me going, we could get some superb recordings, of which one of those I've already shown you.
Can I quickly ask?
Yeah.
Whose eye did you use?
For all these, what I should emphasize is that all these cells, particularly these bipolars and these ganglion cells, they come in a wide variety of shapes and types, and many of them respond in very different ways. The bipolar cells, for example, will respond to complementary colors. If you put a red light on, they'll respond in one polarity. If you put a green light on, they respond in the other. So they show what's known as opponent color coding. They're also very sensitive to small details in the spatial image, so they're critical for seeing fine detail, as is the later cells, as are the later cells. So that's the big take-home message here is that there are a lot of these bipolar cells, they receive their input from the cones, and then the bipolar cells diverge into many different types. And the same story occurs now if we get the last level of this 3-neuron arc. The bottom one, known as the ganglion cells, they get driven by the bipolar cells. So we've got a 3-stage system at minimum. And the ganglion cells down here, sorry, I can't get at them. The ganglion cells, yeah, down here, let's reach that far. They're receiving their input from those bipolar cells. And their bipolar cells are liberating more chemicals. These are, quote, drugs, if you like it. Many of the drugs that have side effects in vision can interfere with some of these chemicals. So it's an electrical and a chemical transmission system. And most importantly, down here, there are many different types of ganglion cells. The upshot is that they also change their response mode, and now this is virtually the end of the story. These ganglion cells here here we show a response at the top to a light flash, and you see there's a series of vertical lines. These are nerve impulses, and they're the first sign they're ever seen in the retina. But what we have now is another code for seeing, and it's a pulse code because it's based on these coals, based on these cells and these pulses, not the size of the signal, which is what we saw earlier with the cones and rods, but they respond with pulses. And so this is now the signal that will be sent up to the brain from the eye. And these things are of many, many different types. And so the big picture here is that we have something known as distributed coding, which is a common computer science technology as well, to say you'll have many, many different cells or channels conveying different information. And the overall signal to be understood has to look at all these different signals. And we have studied these ganglion cells in a lot of detail also, and there are many, many different types. So this is just by way of a summary. This is back to this simple schematic view of the retina. And if you look down, particularly in here, you can see that these processes are going all over the place, connecting other cells in different ways. And each one of them are then generating different classes or types of signals, which finally come out of this ganglion cell. And so, believe it or not, the retina sends all this various types of signals called distributed coding, because the image is distributed across a lot of different channels. And the brain then has to interpret those things and act upon them to finally give rise to vision. But if you don't have these signals coming down through the ganglion cells, again, there is no vision, because everything starts in the retina. And although it's important to be elaborated up here in the brain, the signals from the retina, nevertheless, the retina is where it all starts. And that's why we've studied it so much for so many years. And without patting myself on the back too much, we've had a terrific amount of success over this period, over 40 years actually. I kept doing this research for 42 years at the U, even though many others would graduate and just have graduate students do their research. But I always did it myself. It requires a lot of mechanical dexterity to do this, and a lot of practice. And it also, the last part of my story, I should just add that in my laboratory I've had a number of PhD students who have learned these techniques, and only a few can learn them because they're so difficult. Most graduate students don't want to touch them because they're so difficult. But I had about 5 of these students over the years that I was able to train, and they were able to get to that level of expertise that they could study all these cells in detail. And they went on then to further careers. One was at Stanford and Alabama, another one at Nebraska. These graduate students were a crucial part of my career. But again, unlike others, I didn't just shift everything off to the graduate students. I did the hands-on stuff myself. And let's see, I'd like to make one other point, which actually I'll get back to Henny's point here quickly. This is a list of the many animals that have been used to study the retina. And we won't go down this in detail, except most of them I studied one time or another. All the big ones for me were the walleye, where I was very successful with cones, also with a salamander, tiger salamander, and also the turtle. But anyway, I didn't count all these, but there are probably about 20 here. Some we just tried once to see if it would work at all. And many, many were much inferior to the best ones. So the big 3 in my hands were the walleye, the tiger salamander, and the turtle. Mm-hmm. And we did all sorts of complicated things, particularly with respect to color vision, which was actually sort of my main interest, which I'm leaving out of this talk to keep things simple. And then the last point I wanted to make was just that the support that we received from for scientific research during my career was exceptional. Starting in about, you may remember that the Sputnik appeared in about 1955, and that shook up everybody, and people, including people in the scientific domain. And so grants were set up to start basic research. Basic in the sense that it's not in the clinic, it's not to cure a disease directly, it's all just to understand how the function is working. And they supported, most of their support came from NIH, which is a remarkable institution. It's run into a little difficulty now, which is irrelevant to all they did for 40 years. And actually, in my case, I received a fellowship to do my graduate work at Brown. I received a fellowship to do my graduate work at Johns Hopkins. And thereafter, once I got here in Minnesota, I received research grants for over 40 years to support this research. And it was expensive, it was very difficult to do. It was highly competitive. There are only a handful of people who got money from NIH. And also, the last point, getting back to my students, my graduate students, were also supported by NIH fellowships for their pre-doctoral work. And that made everything possible. So, in sum, this NIH, as it supported basic research in the biological sciences and physical sciences and elsewhere, was really unique. And the emphasis was on basic research. So you wanted to know how things work, not exactly, didn't want you in the eye clinic. They wanted you down in the trenches, as it were, trying to figure out what was really going on. And that's where I spent my career, in the trenches, I guess you would say. But it was a wonderful trench. And it was remarkably successful in my own hands and with my students. And so the students then also were the beneficiaries of the NIH system. And lately it has come under, NIH, as you know, has come under some fire politically, which is just a disaster. In the old days, we never thought about politics with respect to NIH. It was all based on merit. And the competition was intense. And we could only get a grant for a few years, and then we'd have to reapply. So to have support, in my case, for 40-some years was quite a feat. But it made it all possible. So we have to say thanks to the NIH as I end my talk. Thanks.
Can I ask a question?
Sure.
How does the macula fit into the studies that you do? The retina and so forth.
Yeah, the macula contains rods and cones, as you probably know. And they're highly concentrated in the macula, which is this special region, as you know, just in the retina near the center of the eye. We do not record directly from the macula. The macula is basically a specialization of the human eye. But the basic principles of how cones and rods are responding, giving rise to vision, is basically the same. Does that answer your question halfway?
Yeah, kind of, because I have a retina specialist, and he was, well, they call it macular degeneration, but it's basically the retina that's kind of
That's right.
wearing out or whatever.
Yeah,
It's thin, yeah.
Yeah. It's primary. Well, back up a second. The macula has only rods and cones, but particularly cones. And it's just a small segment of the total view. But if that is lost, if the cells are lost there, then the effects are really, really significant, as I'm sure you know. You lose a lot of pattern vision because those cones are not functioning the way they should as individual elements.
Okay.
Is that better?
Yeah, yes, yeah.
Okay.
That helps,
Yeah, yes.
I just wanted to attest to the University of Minnesota in your great research. I have a niece, she's 54, she's a surgical nurse, and she had an eye disease, I won't even quote because I was
Yeah.
out of state, doctor for years, and they saved her eye and
Uh-huh.
they saved her vision.
Yeah.
And it was like 2% of the population had it.
Yeah,
So
yeah.
that because of the university and the research is why she has her eye.
That's right, yeah, by all means, and that's why we did all this research of the sort that I described to finally lead to other things that could restore vision. But it's still restoring vision is a very, very difficult thing to do. And I'll just quickly note that I have a colleague at the U, his name is Gordon Legg, who's an expert in low vision. And he is going to be here in a couple months to give a talk about low vision in particular, including macular degeneration. And he's the expert on that. I just know about the walleye.
Did the walleye eye work better because it was bigger? Because the eye on a fish tends to be bigger, is that?
Yeah, the walleye's eye is pretty big, almost as big as the human eye. Yeah.
Wow.
Yeah. And it also has this sort of grayish cast, if you ever look at the eye.
Like a film.
Yeah, that's right. That's a specialty in the walleye itself. Now, the other side of this coin is that many animals, including many that I've worked on, including the salamander, very, very small. So the walleye's eye is unusually large. Yeah.
Technically, how can lasers help surgeons with
Right.
vision restoration?
Yeah. Well, actually, I've had that done on my own eye. My right eye is not very good anymore. But if you've got a detached retina, as it's called, the retina comes loose. Sometimes that'll just occur in a small part of your eye. And using the laser, you can sort of put the laser image around, let's say it's a circular area where you've lost your vision. You can use the laser to put small spots of light all the way around this area. And sometimes this is loosely referred to as welding.
Yeah, I've heard that said.
And that's basically what it does. So it makes the detachment stop. Otherwise, it just continues. And so that's been very successful for a lot of people.
I've had it done in both eyes, but this
Yeah.
was 3 or 4 years ago. But yeah, I didn't feel a thing.
Yeah, right. Yeah, it's been quite a miracle.
Yeah.
And there are all other aspects of eye research and in the clinic, particularly by the ophthalmologists, which have been quite remarkable over my career in terms of being able to do. But no one has ever been able to do what we and our colleagues have done over the years in terms of looking at these individual cells and actually figuring out exactly what they do. Although there's a lot of theorizing about this, as you might expect. This is all just a hypothetical theorizing about how the connections might be made. And it's super, super simple because every class of cells, the bipolars, the ganglion cells, there are many different types. For example, there are ganglion cells that will respond to something that moves in one direction and not at all to the other. They're called directionally sensitive cells. And there are a lot of color sensitive cells that I haven't talked much about in the retina. That's all laid down also even in a number of cells which are horizontal cells, which also show this color opponent coding between complementary colors. So I've avoided color vision on purpose, not because I'm uninterested, because I spent most of my life trying to figure it out, but I never did figure it all out. Yeah.
What's the structural connection between this and the optic nerve?
Actually, we can see it in this slide. You can see the ganglion cells down here. Now, they have very thin processes or fibers that come from them that are shown down at this level, and it says NFL, which is nerve fiber layer. And so these are all destined to the brain. And they define what is usually referred to as the optic nerve, which is actually something you can see just with the naked eye. But again, there are thousands to millions of these, and they're all sending different sources of signals, and it all has to be put together to give us a coherent picture of vision and what's happening in the world, which is still a miracle, as Shakespeare was still on target when he referred to the miracle of an eye.
It's a super complex item, right, or thing, whatever you want to call it.
Is
eye
it what?
Super complex. Sounds the complexity of it.
Yeah. Nobody, well, maybe somebody would have guessed, but it wasn't until this research that we started with many others in the 1960s, 70s that we got a clue as to what these things were doing. And the answers were very different than what people had theorized would be the case. Yeah.
Has there ever been yet a total transplant of an eye?
No, not that I know of.
Parts of each of the eye have been, like the,
Yeah.
what different things can they actually transplant?
Actually, I'm not too familiar with this, but I can say very clearly and importantly, you cannot transplant a retina. And also, there's been a lot of work in the last 20 years trying to create artificial retinas, or implants, as they're called. And there have been various interesting attempts to do this, ever so complicated. And actually, they've been very, very minimally successful. So we're a long way from being able to do that.
Now, does the retina on the outside of the eyeball, or is it inside of it, or how does it work?
It's on the inside,
Okay.
just as,
I always thought it was on the outside.
well, let me get back.
The back.
Okay.
We'll get there eventually.
I'm trying to be
There you go.
Yeah. So now we're looking at this slide, and the retina is that cross-hatched layer on the inside of the eye. So it's all on the inside of the eyes. So you take the eyeball, and then you have a very thin film of nerve cells, and that's the retina over the entire area. Yeah.
And the,
I was just wondering, did you find out how many rods have to be there to have the eye functioning? And have they ever tried to do rod replacement?
Yeah.
Which is like replacing
Yeah.
retina.
Rod replacement, I don't think has even been tried. Would be so impossible. Now, the other thing, what was the first
How many?
question?
How many rods?
Do you need to see?
Oh,
Millions?
well, they range from about 5 to probably thousands.
Yeah.
And one of the most interesting early finds on human vision is that if you can excite just a few number of rods, it's possible for you to see a very, very weak light flash. But the interesting part of that finding is it indicates that these cells are super, super sensitive. So if you remember anything about the physics of light, it consists of a number of light particles, which are called quanta. And at the very, very dimmest lights we can see, there are only a few quanta around. In this room now, there are literally thousands or millions with bright lights. But anyway, when you get just a few of these photons or quanta active, it can produce a just vague perception that's astounded people, both in vision and throughout the world, that the eye is so super, super, super sensitive. I didn't talk about that either. At least I don't think so. So I certainly thank you for your attention. I've tried to keep it straightforward.